Okay, guys, so this chapter is an overview on chemical principles that are typically associated with biology. Again, I really just want you guys to sort of review this chapter. Um, don't spend too much time on this, as it's just sort of like an overview. So matter is anything that has mass and can occupy space, and there are three states of matter: solid, liquid, and gas. Energy, this is the capacity to do work or put matter into motion. And we typically have two forms of energy: kinetic energy and potential energy. Kinetic energy is actually energy that's in motion or action, and potential energy is stored or inactive energy. Now, energy can be transferred from potential energy to kinetic energy. And there are different forms of energy. You have chemical energy, electrical energy, mechanical, and then radiant or electromagnetic. Now, energy can be converted from one form to another. And what we've learned is that the conversion of energy is typically a very inefficient process. And a lot of times we lose energy as heat, which is not usable, and it's also unstable. The elements. Elements are matter is going to be composed up of elements. They cannot be broken down into simpler substances by ordinary chemical methods. And each has unique properties. You have physical properties, which are detectable with our senses or are measurable. And then you have chemical properties. This is how atoms will interact or bond with one another. Atoms, these are unique building blocks for each element. They give each element its physical and chemical properties. The smallest particles of an element are with properties of that element. Atoms are the smallest particles of an element with properties that are of that element. An atomic symbol is one or two letter chemical shorthand for each element. Okay, when we look at the body, the human body, there are four elements that will actually make up 96.1%, and that is carbon, hydrogen, oxygen, and nitrogen. You should know that. Okay. 9 elements will actually make up the remaining 3.9% of body mass. And you'll see these throughout the course, these terms or these elements: calcium, phosphorus, potassium, sodium, chlorine, magnesium, iodine, iron. So you'll see these throughout, okay? Obviously, there's a lot more elements on the periodic table of elements, some which can be part of the human body, but they are often in what we call trace amounts. So we also have 11 elements that will make up less than 0.01% of body mass, and many are part of or activate enzymes. And you can see some of those here, such as copper, manganese, silicon. So atoms are composed of subatomic particles: protons, neutrons, and electrons. Protons and neutrons are found in the nucleus, and electrons will orbit the nucleus in an electron cloud. Almost entire mass of the atom is found within the nucleus. And the nucleus, again, will contain neutrons, which carry no charge, and protons, which carry a positive charge. And here you can see a typical element. This is for helium. Now, making molecules, most atoms will chemically combine with other atoms to form molecules and compounds. So a molecule is two or more atoms bonded together, the smallest particle of a compound with specific characteristics of that compound. So the compound is two or more different kinds of atoms bonded together. Most matter exists as mixtures, two or more components physically intermixed. And there are three types of mixtures that exist: solutions, colloids, and suspensions. We have what are homogeneous mixtures. Homogeneous means that they are made up of the same substance. Most are true solutions in body. Gases, liquids, solids are dissolved in water. The solvent, this is the substance that's present in the greatest amounts. It's usually a liquid, which is usually water in the human body. The solute, this is present in smaller amounts. So if glucose is dissolved in blood, glucose is a solid, blood is a solvent. Concentration of true solutions can be expressed as the percent of the solid in total solution. And again, assume that it's going to be water. So parts of the solid per 100 parts of solvent. Typically, you'll see this if you have ever had lab work done and they're looking at something, it'll typically be milligrams per deciliter. You might even see molarity or moles per liter as far as the expression is concerned. Colloids, or AKA emulsions, these are heterogeneous mixtures. For example, this would be the cytosol, which is what we find inside the solution inside the cell. Large solid particles do not settle out. Some undergo solid to gelatinous transformations. And then suspensions, these are heterogeneous mixtures such as blood, large visible solids, which do settle out. And here you can see the difference between a solution, colloid, and a suspension. So mixtures, there is no chemical bonding between components. They can be separated by physical means, such as straining or filtering. And a mixture could be heterogeneous or homogeneous. Heterogeneous, again, means composed of more than one type. Homogeneous is just a single type. Compounds, chemical bonding between components can be separated only by breaking bonds, and all are homogeneous. So what are chemical bonds? Chemical bonds are energy relationships between electrons of reacting atoms. There are three major types. We have ionic bonds, covalent bonds, and hydrogen bonds. Ionic bonds is when you have the transfer of a valence shell electrons from one atom to another forms ions. And the attraction of opposite charges results in an ionic bond. So here you actually have the formation of an ionic bond where you have the transfer of ions from one molecule to another. Sorry, one atom to another. Most ionic compounds are salts. Covalent bonds, these are formed by the sharing of two or more shell electrons. This allows each atom to fill its valence shell at least part of the time. So the difference between an ionic bond and a covalent bond is ionic bond, you have the donation and the reception of an electron. For covalent bonds, they are actually sharing the electrons, as you can see here. Hydrogen bonds, these are attractive force between electropositive hydrogen of one molecule and an electronegative atom of another molecule. This is not a true bond. It's common between dipoles such as water. And it also acts as intramolecular bonds holding a large molecule in a 3D shape. Typically, I think I mentioned this. Nope, I did not. So typically, hydrogen bonds will exist between hydrogens of an oxygen and hydrogens of a nitrogen. So you'll typically see oxygens and nitrogens involved in the formation of hydrogen bonds. So chemical reactions, this occurs when chemical bonds are formed, rearranged, or broken. It's represented as a chemical equation using molecular formulas. And chemical equations will contain both reactants and products. Okay, and one rule of thumb is that whatever goes into an equation has to come out of the equation. Typically, reactants are on the left-hand side of an equation. Products are on the right-hand side. And the arrow is typically indicative of an enzyme that's facilitating that reaction. So different types of chemical reactions. We have synthesis reactions where you're going to form things, decomposition reactions where you'll break things down, and exchange reactions where you'll take one thing and convert it into something else. So in this reaction, this is a synthesis reaction. You have A plus B yields to AB. This is what we typically call an anabolic reaction, and synthesis reactions typically involve the input of energy. So it's going to cost energy. And here you can see an example. Decomposition reactions, this is when you take a larger molecule and you break it down into two smaller molecules. This involves breaking a bond, okay? And this is what we refer to as a catabolic reaction. And this will actually lead to the release of energy. So your anabolic or your synthesis reactions are going to require energy because you have to form bonds. Your catabolic or decomposition reactions are going to release energy because you're breaking bonds. And an exchange reaction, this is when you have AB plus C leads to AC plus B. Okay. They involve both synthesis and decomposition. Bonds are both made and broken. So energy-wise, it's kind of a wash. In the body, we have what are referred to as oxidation-reduction reactions or redox reactions. These are decomposition reactions. These are reactions in which food fuels are broken down for energy. They also exchange reactions because electrons are exchanged between the reactants. And when we break down glucose, essentially you're going through a bunch of redox reactions to accomplish this. But the thing that happens here is the oxidation is the release of electrons. The reduction is the gain of electrons onto something else. And whenever you oxidize a molecule, you have to reduce another molecule. These two things go together. All chemical reactions are either exergonic or endergonic. So exergonic reactions, you have a net release of energy. So this would be catabolic or oxidative, because you're releasing energy under both those terms. Endergonic reaction, you have a net absorption of energy, and this would be anabolic or a reductive reaction. So all chemical reactions are theoretically reversible, meaning that you can go either way, but that's not always 100% the case in the human body. Chemical equilibrium occurs if neither a forward nor reverse reaction is dominant. Many biological reactions are essentially irreversible, and due to energy requirements and due to the removal of products. That's why they can be irreversible. The rate of chemical reactions is affected by temperature. If you increase the temperature, typically you'll increase the rate of a chemical reaction. Concentration, if you increase the concentration, you'll increase the rate. Particle size, if you decrease the particle size, this is inversely proportional, where you would actually increase the rate. And then catalysts. Catalysts are biological, are going to essentially lower the activation energy of a chemical reaction, and you typically will see an increase in the rate. Enzymes, which is a term that we use in the human body and actually all living cells. Enzymes are essentially catalysts. They're biological catalysts. Okay. Oops. Okay, so biochemistry, this is actually going to be the study of chemical composition and reactions of living matter. All chemicals either will exist as organic molecules or inorganic. And an inorganic compound would be something like water, salts, many acids and bases. They do not contain carbon. Organic compounds, these are carbohydrates, fats, proteins, nucleic acids. They do contain carbon, usually large. They are usually very large. And they are made up of atoms that are covalently bonded to make these large molecules. Both are equally essential for life. So water in living organisms, this is the most abundant inorganic compound, 60 to 80% of volume of living cells. It is also the most important inorganic compound due to the water's properties. It has a high heat capacity. Which absorbs and releases heat with little temperature change. It also prevents sudden changes in temperature. It has a high heat of vaporization. This is evaporation, requires large amounts of heat, and it's useful as a cooling mechanism. Water is also a polar solvent properties. It is going to dissolve and disassociate ionic substances. Will also form hydration layers around large charged molecules such as proteins, and it's the body's major transport medium. Water is reactive, which is necessary part of hydrolysis and dehydrolysis synthesis reactions. It also has cushioning, which protects certain organs from physical trauma, such as cerebral spinal fluid, which protects the brain and spinal cord. Okay, an inorganic compound would be salts, ionic compounds that dissociate into ions and water. They contain cations other than hydrogen and anions other than hydroxide. Common salts in the body are sodium chloride, calcium carbonate, and potassium chloride. Acids and bases. These are electrolytes. They ionize and dissociate in water. Acids are proton donors, where they release protons in solution, and bases are proton acceptors, where they take up protons from a solution. Some important acids are hydrochloric acid, which we'll see in the stomach, bicarbonate, which is the H2CO3. An important base would be bicarbonate, which is HCO3, as well as ammonia. So when we look at pH, okay, we have a pH scale, and it ranges from 0 to 14. And then what a pH scale is, it's actually logarithmic, which means when you move from one number to the next, it's actually 10 times. It's on a tenfold. That's what a log scale is. Log scale means that it's on a basis of tenfold. So an acidic solution is anything between 0 and 6.99. Neutral solution is 7, which is pure water, which is not something we typically find, okay? When we talk about water, it's actually not pure water. And then an alkaline solution would be anything from 7.01 to 14, which is a basic solution. Here's a pH scale. It also has some common things that you guys are familiar with, such as milk, wine, lemon juice, bleach, ammonia, and you can see where they typically fall on the pH scale. So neutralization, this results from mixing acids and bases together. Displacement reactions occur forming water and salt. So if you actually mix an acid and a base together, and you're essentially going to end up with salt water. That's what a neutralization reaction is. So if you took pure HCl, which is hydrochloric acid, which is the strongest acid, and you took pure sodium hydroxide, which is the strongest base, what you would end up with is water and sodium chloride, which is as a result of an exchange reaction or a neutralization reaction, giving you salt water. pH change interferes with cell function and may damage living tissue. Even a slight change in pH can be fatal. For example, blood pH is somewhere between 7.35 and 7.45. We'll get to that a little bit later on. Any variation in blood pH could be very, very bad. It's fatal if you go below 7.35 or above 7.45. The body oftentimes has buffer systems. These buffers will resist abrupt and large swings in pH. They can also convert strong acids or bases into weak ones. Okay, our organic compounds. These are going to be your carbohydrates, lipids, proteins, and nucleic acids. They are very unique to living systems. And they most likely will contain carbon. Many organic compounds are polymers, which means they are chains of smaller units called monomers, which are building blocks. And they are synthesized by dehydration reactions, and they are broken down by hydrolysis reactions. And here you can see the dehydration reaction, which would be a catabolic reaction. No, I'm sorry. Dehydration reaction would be an anabolic reaction where you're essentially synthesizing larger molecules from smaller ones. A hydrolysis reaction would be a catabolic reaction where you're taking a larger molecule and you're splitting it into two smaller ones. So our first organic molecules, carbohydrates. This is sugars and starches. They do form polymers. They contain carbon, hydrogen, and oxygen in the typical formula of CH2O, and then, N to the N power. So for example, if you had N was 6, it would be C6H12O6. Okay, so for every carbon and oxygen, you'll have 2 hydrogens. That's what that's saying. There are 3 classes. You have monosaccharides, which is 1 sugar, disaccharides, which is 2, and polysaccharides, which is many. The functions of carbohydrates is that it's oftentimes a major source of cellular food, such as glucose. And it can also be found as a structural molecule. Ribose sugar, which is found in RNA, deoxyribose sugar, which is found in DNA, for example. So your monosaccharides, these are your simple sugars that contain 3 to 7 carbon atoms. And again, it follows that CH2O to the N general formula. Monomers of carbohydrates, this is what will form those. And important monosaccharides are your pentose sugars, which is your ribose and deoxyribose. Those are 5 carbons. And your hexose sugars would be examples such as glucose, which is 6 carbons. And here you can see some examples of those monosaccharides. Disaccharides, these are double sugars. They're too large to pass through cell membranes. And important disaccharides are things like sucrose, maltose, and lactose. You can see some of those here. Polysaccharides, these are polymers of monosaccharides. There are several important polysaccharides, such as starch and glycogen. And they are not very soluble in water. And an example would be that of glycogen, which is what we refer to as starch. Lipids, these contain carbon, hydrogen, and oxygen as well, less than in carbohydrates, and sometimes you'll see phosphorus. Lipids, unlike carbohydrates, are insoluble in water, and there are many different types. There are triglycerides or neutrofats, phospholipids, steroids, and econocides. So triglycerides or neutrofats, they're called fats when solid and oils when liquid. They're composed of 3 fatty acids, chains bonded to a glycerol molecule. And the main storage of triglycerides is energy storage, insulation, and protection. And here's an example of a triglyceride where you have 3 fatty acid chains and a glycerol molecule. Saturated fatty acids, this is going to contain a single covalent bonds between the carbon atoms. Unsaturated means you'll have 1 or more double bonds. Unsaturated are referred to as your heart-healthy fatty acids, and these are your plant oils and olive oils. Saturated ones are butter, animal fats. Trans fats, these are modified oils. They're typically unhealthy. And then your omega-3 fatty acids, these are heart-healthy. Phospholipids, these are modified triglycerides. You have glycerol plus 2 fatty acids and phosphorus containing group. This will have a head and a tail region with different properties. Typically the head region or the phosphorus containing group, this is polar, which means it likes water. And then the tail region, which is 2 fatty acid chains, is nonpolar and it does not like water. Steroids. Steroids are interlocking 4-ring structures. Examples of steroids would be cholesterol, vitamin D, your steroid hormones, which we'll talk about a little bit later on in this course, and your bile salts, which we'll talk about in AMP2. Most important steroid is cholesterol. It's important in cell membranes, vitamin D synthesis, steroid hormones, and bile salts. Here's the simplified structure of a steroid. And this is actually cholesterol. The eicosanoids, there are many different ones. They are derived from fatty acids, such as arachidonic acid. Most important eicosanoid is prostaglandin, which has a role in blood clotting, controlling blood pressure, inflammation. So you'll see some of these play a role as well. Other lipids in the body, we have our fat-soluble vitamins, which are A, D, E, and K. And then we also have lipoproteins. Okay, our third category is proteins. And with the proteins, They will contain carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur and phosphorus. Proteins are polymers. They're made up of amino acids, and there are 20 different amino acids that we typically find. Amino acids are joined by covalent bonds called peptide bonds. They will contain an amine group, and they will also contain a carboxyl acid group. They can act as either an acid or a base, and all amino acids are identical except for the R group. The R group, which is indicated in green here, is what changes from one amino acid to the next. And here, looking at a typical base of amino acid, the COOH right here, this is your acid. The NH2 here, this is the amine group. This will always just be a hydrogen, and then the R group is what varies from one amino acid to the next amongst the 20. So you have two amino acids that come together through a dehydration reaction. They form a peptide bond, and then you start to build a protein. And here you can see, excuse me, primary structure for protein, which is just all the amino acids linked up together. As these amino acids start to come together, because they all have different R groups, and all those R groups have different chemical properties, what you're going to start to see is some of those R groups interacting with each other, which will start to fold the protein. And you can either fold it into a helical structure, which is shown here, or what we call beta sheets. Beta sheets are like the pleated lines sort of motif. And then it will continue to fold on itself until it folds into a very unique structure, which is what we call a tertiary structure. A quaternary structure is really two proteins that have formed bonds with each other. So you essentially have two or more tertiary structures that have formed bonds with each other. Hemoglobin, which is a molecule that we'll talk about in AMP2, actually is four separate proteins that are bonded together. Okay. Let's see. Proteins can be denatured, which means that they can unravel. What denaturation means is that the bonds holding the protein together are being broken down and the protein unfolds or unravels. If a protein unfolds from its structure, it loses its function. One of the things with proteins that's a bedrock rule is that structure always dictates its function. Molecular chaperones, these are globular proteins that will ensure quick and accurate folding of proteins that are being synthesized by the cells. They will also help to get those proteins across membranes. And they will also help to facilitate the breakdown of damaged proteins, because you don't want damaged proteins just lingering around either. Enzymes are proteins. We'll talk more about these in AMP2 and their functions. I'm not going to get too much into the characteristics of enzymes here. We'll get into that a little bit later. Okay. So the final organic compound is nucleic acids, and this is your DNA and RNA. This is the largest molecules in the body. They contain carbon, oxygen, hydrogen, nitrogen, and phosphorus. They're also polymers of nucleotides. So DNA will utilize four nitrogen bases: adenine, guanine, cytosine, and thymine. Adenine, guanine are your purines, cytosine and thymine are your pyrimidines. There's a base pairing rule. The base pairing rule is adenine always pairs up with thymine, and guanine will always pair up with cytosine. Double-stranded helical molecule is typically what we think of when we think about DNA. It's a double helix. The pentose sugar is deoxyribose. DNA provides the instructions for protein synthesis. It replicates before cell division, ensuring genetic continuity. And here you can see a DNA molecule, and it's sort of what we call a ball and stick model there. RNA, on the other hand, there are four bases. You have the adenine that we see in DNA, the guanine, the cytosine, but instead of thymine, we have uracil. So wherever you had a thymine in DNA, it would end up just being a uracil in RNA. There are three main types of RNA that carry out DNA orders for protein synthesis, and that is messenger RNA, transfer RNA, and ribosomal RNA. And we'll talk about that a little bit later on in the course as well. ATP, which is the major energy molecule that we associate with in cells, is also a nucleotide. Chemical energy in glucose is captured in this important molecule. It directly powers chemical reactions in cells. The energy form, immediately usable by all body cells. The structure of ATP is an adenine-containing RNA nucleotide with 2 additional phosphate groups. So here you can see the adenine, ribose sugar, and then we have 3 phosphates bonded together in ATP. The energy formed by this bond between 2 phosphates is where all the energy is. It's not actually the molecule, it's the bond between the phosphates. So functions of ATP, you can have phosphorylation. This is where you get the terminal phosphates are enzymatically transferred to and energize other molecules. And then you can also have ATP involved in transport work, mechanical work, as well as chemical work, meaning it can fuel transporters in cell membranes. It can lead to muscle contractions. Chemical work would be it fuels enzymatic reactions. Okay. So again, this was an overview of chemical properties. We started off with some atoms and matter, talked about different types of chemical reactions, then got into inorganic compounds and then our organic compounds. I really want you guys to focus on the organic compounds. Make sure you know the differences between carbohydrates, lipids, proteins, and nucleic acids. That's very important, especially as we start talking about a lot of these. Okay. All right. Have a good one.